In this PowerPoint, I'll be going through types of transport across the plasma membrane. In the area of study 1 outline, we can see this under crossing the plasma membrane. So the modes of transport of soluble substances across the plasma membrane, including simple diffusion, facilitated diffusion, osmosis, and active transport. Diffusion occurs when particles move from an area of high concentration to an area of low concentration. The particles are said to have moved along the concentration gradient, or moved down their concentration gradient. We can see this in the diagram on the right, as we see solute particles in a high concentration in the corner of the beaker. Over time, they will diffuse throughout the solution until they reach equilibrium, or until they're evenly spread throughout the solution. Diffusion is a passive type of transport, meaning that it doesn't require energy to occur. There are two types of diffusion across the plasma membrane, either simple or facilitated. Simple diffusion occurs when the plasma membrane is permeable to the particular solute molecules. When the solute molecules can diffuse across a membrane, they can diffuse from a higher concentration to an area of low concentration. In other words, they diffuse down their concentration gradient or along their concentration gradient. On the diagram shown, you can see that as time progresses from left to right, the particles diffuse from the top to the bottom, from an area of high concentration to an area of low concentration. They'll continue to diffuse until they reach a state of equilibrium, which is shown here. Three factors can affect the rate of diffusion across a membrane. The first one is concentration. The greater the difference in concentration, the faster the rate of diffusion. Second one is temperature. The higher the temperature, the higher the rate of diffusion. This is why sugar will dissolve more quickly in warm water compared to cold water. The last is particle size. The smaller the particles, the faster the rate of diffusion. This is like if you had a cube of sugar and you tried to dissolve it, it would actually dissolve much more quickly if you crushed the sugar cube into individual sugar granules. The next type of diffusion is facilitated diffusion. So for particles that are unable to cross because the membrane is impermeable, so this includes ions, large molecules, or polar molecules, they require some help from a protein. First, we have protein channels. These allow these particles to move from an area of high concentration to an area of low concentration. Without the use of these protein channels, they would not be able to cross the membrane. So when proteins help the movement down the concentration gradient, it is known as facilitated diffusion. It's much more quick than simple diffusion. It is also a passive type of transport, meaning no energy is required. They are still diffusing down or along their concentration gradient. There are two types of membrane transport proteins that help with facilitated diffusion. First, we have channel proteins, which are shown in the diagram here. And we also have carrier proteins, which are shown here. Membrane proteins are specific for particular particles, meaning that a specific particle will have a specific protein channel that enables it to cross the membrane. The transport of one protein can be inhibited by the presence of another that uses the same transport system. This could be channel protein blockers and would inhibit one particular particle to cross. This can be quite harmful to the cell. So to summarize the three types of passive transport known as diffusion, we have simple diffusion, where it crosses the membrane from an area of high concentration to an area of low concentration without the use of a protein. We have channel protein with a pore that enables particles to transport across the membrane from higher concentration to lower concentration that would not normally be able to cross. Lastly, we have a carrier protein. A carrier protein is able to help transport a particle across the membrane, also from an area of high concentration to low concentration, but it can only transport them in one direction at a time. Next, we have a special case of diffusion known as osmosis. In osmosis, water particles move along the concentration gradient from an area of high water concentration or dilute to an area of low water concentration. In other words, the water diffuses down the osmotic gradient. The pressure causing the water to move along this gradient is called osmotic pressure. We can see on the diagram on the right that on the left side of the differentially permeable membrane, we see a high concentration of water particles. And on the right, we can see that there is some water particles, but there are also some sucrose molecules. Normally, we would expect that the sucrose molecules would diffuse by simple diffusion from the right side to the left, but as they are unable to diffuse across the membrane, the water will diffuse from the left side to the right to reach equilibrium. In these diagrams, we can see the process of osmosis. In the first beaker, we can see a beaker filled with pure water, so it's got 100% free water molecules. In the second beaker, we can see a sugar solution. So sugar has been dissolved in the water solution, so there are hydrated sugar molecules. This means that the water molecules are reacting with the sugar molecules. There are still some free water molecules. If we put the two solutions together and separated them by a semi-permeable membrane, like in diagram 3, we can see that the net movement of water will be from the left side to the right side of the membrane. This would reach equilibrium. What you'd expect is that the water level on the right side of the membrane would increase and the water level on the left side of the membrane would decrease due to the net diffusion of water. In this diagram, we can see a beaker separated into two sides, A and B. In the first diagram, we can see that A and B are separated by a selectively permeable membrane. Side A has a higher concentration of sugar molecules. If we see diagram 2, we can see that the water level on side A has increased. This is because the net diffusion of water was from side B to side A. As sugar molecules couldn't diffuse across the membrane, water diffused from a higher water concentration to a low water concentration in order to reach equilibrium and balance the concentrations between the two sides. There are three key words that we use to compare the solute concentrations between two solutions. When we talk about the three types of solutions, we refer to the external solution. The external solution can either be isotonic, hypertonic, or hypotonic. If the external solution is isotonic, that means that the solutions being compared have equal concentration of solutes. There is no net flow of water between the two solutions, and you would not expect a volume change. If the external solution is hypertonic, that means the solution has a higher concentration of solute, or in other words, a lower concentration of water. Think hyper as in higher. Lastly, we have a hypotonic solution. If the external solution is hypotonic, that means that this solution has a lower concentration of solute, or a higher concentration of water. Think hypo as lower. On this slide, we can see what happens to a red blood cell if it's placed into each of these three solutions. Remembering that the cell membrane of a red blood cell is permeable to water. If we put these red blood cells into an isotonic solution, as in the first beaker, you would expect that there would be no change in the cell size, as there would be no net movement of water into or out of the cells. If the cells were placed into a hypertonic solution, you'd expect that the net movement of water would be out of the cell to balance out the concentration. As a result, the cells would shrink. Lastly, if we put the red blood cells into a hypotonic solution, we would expect that the net movement of water is into the cells. As a result, the cells would swell and potentially eventually burst. Here we see red blood cells compared to plant cells in each of these three types of solutions. As we know, in an isotonic solution, the red blood cell would not see a change in size, The same would happen for a plant cell. The plant cell would retain the same size and shape due to the equal diffusion of water into and out of the cell. While we place the red blood cell into a hypotonic solution and the cell would swell and burst, due to the cell wall in a plant cell, it would swell, but it wouldn't burst due to the cell wall keeping its pressure and shape. In the last diagram, we see hypertonic. You would expect a red blood cell to shrink. The same would happen to the inner part of the cell of a plant cell, including the cell membrane. However, the cell wall would still retain its shape. Plants with high internal fluid pressure are called turgid, or they have a high turgor. What I recommend doing now is to stop and answer these three questions. What is a concentration gradient? What does it mean for a molecule to diffuse down a concentration gradient? And complete the concept map about passive transport using the following words and phrases: osmosis, energy, facilitated diffusion, the movement of molecules from high to low concentrations, and diffusion. Next, we have active transport. Active transport is different from passive transport as it requires the use of energy by the cell to transport particles across the membrane. The form of energy used by the cell is known as ATP. Active transport occurs with the use of carrier proteins, and it is a movement of substances against the concentration gradient. The properties for active transport are the same as facilitated diffusion. First, we have selectivity. So this means that some substances are transported while others aren't. If we look at the diagram on the right, we can see that substance A alone is not transported. We can conclude this because even if the concentration of the substance increases, there is no increase in the rate of transfer through the channel. Next, we have saturation. So there's no increase in the rate of transfer when all transport proteins are occupied. So we can see this is that if all protein carriers in the membrane are occupied, then there will be no increase in the rate. We can see this, that each of the graphs for C, B, and B in the presence of C all flatten out at a certain point. Lastly, we have competitive inhibition. So one substance can inhibit or block the transport of another substance by using the same transport protein. We can see this for B in the presence of C. So when C is present, there is a lower rate of transfer for B due to this competitive inhibition of transfer. So how does active transport relate to the overall function of the cell? We can see here examples of exocytosis and endocytosis. So when large molecules in fluid are moved in and out of the cell, these require energy. So these two types are types of active transport. Exocytosis is a movement of substances out of a cell. We can see in the diagram that a transport vesicle, for example, might be containing digestive enzymes, will fuse with the plasma membrane, and then the junction between the membrane and the vesicle will break down, and those enclosed materials, so those digestive enzymes, will be released on the outside of the cell. Below it, we can see endocytosis. Endocytosis is the movement of substances into the cell. The particles are enclosed by the membrane, which then pinches off at the top to form a vesicle. These might fuse with lysosomes so that contents can be digested for use by the cell. Then we have pinocytosis. Pinocytosis is the entry of extracellular fluid and proteins or sugars. Phagocytosis is the entry of large particles such as bacteria and cell debris. In this diagram, we can see a summary of the two different processes taking place. On the left side, we can see endocytosis. So we look down here, we can see that some particles are enclosed by the plasma membrane into a vesicle. The vesicle might combine with a lysosome, which contains digestive enzymes. These break down cell parts so they can be used in other parts of the cell. So here we can see the enzymes, and they combine with the vesicle, and it's digested to bring apart the lipids. The lipids are then transported to the Golgi apparatus, where they might be used and transported somewhere else in the cell. On the other side, we can see exocytosis. Exocytosis begins up the top. So if we start with the proteins are taken to the Golgi apparatus. Then they might be modified for the proteins and lipids and packaged into a vesicle. The vesicle is then transported to the plasma membrane where it fuses, and then those are secreted on the outside of the cell. Now that you've reached this point, I recommend answering the following questions. How is active transport different than simple diffusion and facilitated diffusion? The prefix exo means out of, and the prefix endo means taking in. How do you think these meanings relate to the meaning of exocytosis and endocytosis? For question 3, what is the process shown in figure A, and what is the process shown in figure B? So let's summarize. We have two main types of transport for moving substances across the plasma membrane. We have passive transport. This moves solute particles from an area of high concentration to an area of low concentration. They are transported down or along their concentration gradient, so there is no energy required. This includes simple diffusion, facilitated diffusion, and osmosis. Remember that facilitated diffusion requires the use of protein channels or protein carriers. Then we have active transport. In active transport, particles are transferred from an area of low concentration to an area of high concentration, so from an area or against their concentration gradient. As a result, this requires energy. Active transport requires proteins embedded in the membrane, and these have particular properties which make them selective to the particular particles that they transfer. In this diagram, we can see the different mechanisms of solute transport being summarized. We can see simple diffusion, the use of an ion channel, and facilitated diffusion are all types of passive transport. No energy is required because they are diffusing from an area of high concentration to an area of low concentration. In comparison, we have active transport. Active transport transports materials from an area of low concentration to an area of high concentration. As it moves against the concentration gradient, energy is required. The energy used is in the form of ATP. As a summary task, I recommend making a foldable summarizing the following types of transport: simple diffusion, facilitated diffusion, osmosis, and active transport. This will be really helpful when it comes to revision. So let's look at our key ideas so far. Simple diffusion moves dissolved substances across the plasma membrane down their concentration gradient and requires no input of energy. Osmosis is a special case of diffusion, being the movement of water across the plasma membrane down its concentration gradient. This is known as the osmotic gradient. Facilitated diffusion moves dissolved substances across the plasma membrane down their concentration gradients, but this movement occurs through the involvement of transport proteins, either channel or carrier proteins, and requires no input of energy. Active transport moves dissolved substances across the plasma membrane against the concentration gradient, a process that can occur only via the action of protein pumps. Active transport requires an input of energy that commonly comes from ATP. This is catalyzed by the ATPase enzyme that is part of some protein pumps. Endocytosis is the bulk transport of material into cells. If solids are being moved, the process is termed phagocytosis, and if liquids, the process is termed pinocytosis. Exocytosis is the bulk movement of materials via secretory vesicles out of cells.